underneath the light microscope. So again, here's your textbook's depiction of prophase, here's prometaphase, and now we're getting ready to move into the next stage, which is called metaphase. And as you can see, metaphase is going to have some very nice distinct changes for us to see. This is a really neat looking slide. This is a slide that was taken using fluorescent microscopy. And fluorescent microscopy makes use of very sort of spectacular dyes. Each dye is intended to stain a different part of the cell. So, for example, in this case, you're looking at a single cell, but you can't see the membrane. You can't see the cytoplasm. That's all in black. But you can see some of the specifically stained components inside the cell. The blue material, for example, is the nuclear material, the chromosomes. The red material is the spindle fibers. You can see them at each end. And the yellow material is the centrosome that contains the centrioles. So when I look at this slide, I can tell, number one, that we're in mitosis because I can see the chromosomes, and number two, that we're in prometaphase. The chromosomes are no longer enclosed in a nice, neat nuclear envelope. Instead, what we see is they're disorganized and they're just sort of free in the cytoplasm. That's the key that this is prometaphase. Now, in this slide, we have another section of a plant. This happens to be onion cells, and these are the same types of cells we'll be looking at in lab on Thursday. So this is an individual cell here, and this is an individual cell here. Here are a couple more cells. Here's a cell. Here's a cell. Most of these cells have very homogeneous looking nuclei. These are all cells in interphase. They are either in G1 or in S or in G2. We just can't tell which. This cell is different, and so is this cell. Now these are labeled early prophase and late prophase, but for us, I want you to learn this one as prophase and this one as prometaphase. Yes, this is an example where we actually caught prometaphase occurring, but again, that's typically not the case, and we may not get to see any of these types of nuclei on Thursday. So we know this is prophase because it still has a nuclear envelope, a nice smooth oval outline. It's got condensed chromosomes. We can see them. And even though we can't see it, the spindle fibers are forming out here somewhere. This one is now prometaphase because, as you can see, the chromosomes are no longer tucked into a nice oval nuclear envelope. They've been released. The spindle fibers are all in place. We just can't see them. And that brings us to metaphase. And metaphase is one of the really neat ones to see underneath the microscope because it has a very unique appearance. During metaphase, the chromosomes are going to line up in the center of the cell. And the kinetochores, those little black dots that we saw in the drawing, are actually going to attach along what we call the metaphase plate. Now, the metaphase plate is not an actual visible structure for us. It's just a line, if you will, in the center of the cell where the chromosomes are going to line up with the kinetochores along that line. If you look into your textbook, you'll see it drawn like this. So they're lining up basically head to tail, head to tail, head to tail, all the way across this invisible metaphase plate. Spindle fibers are attached to the kinetochores. The nuclear envelope is gone, remember, and the centrosomes and centrioles are still at the poles of the cell. This is what it would look like using fluorescent techniques. Again, you can't see anything out here. This black area is what's going on in the cytoplasm. But you can see these beautiful greenish colored spindle fibers. You can see The centrosome area would be right here with the centrioles, and the spindle fibers are growing out in all directions, but particularly up towards these red chromosomes. And although it doesn't look like a nice perfect straight line like it looks like in your textbook, you can certainly get the idea that these chromosomes have lined up along some sort of a plate in the center of the cell. This slide shows an onion cell that's in metaphase, and this is very similar to what you'll see in lab on Thursday. Again, most of the cells on this slide are in interphase. The nuclei look very just reddish pink. You can see visible nucleoli here and here and here and here and one here, but all of these cells are in plain old interphase, G1, S, or G2. But look at this cell. Something very unique is going on in this cell, and that's metaphase. Again, it doesn't look as perfect as it looks in drawings, but I think you can see that there's a straight line of chromosome material right here, and what looks like little spiky legs coming off of it. I think this looks something like a spider. That's what I look for when I look under the microscope at these figures, these mitotic figures. This looks like a spider to me with its legs sticking out in all directions. And again, what's happening is the kinetochores are lining up along the center of the cell, and the ends of the chromosomes are sort of hanging off like the spider legs. Here's another picture of an onion cell in metaphase. This is a really nice one because you can imagine that you're seeing spindle fibers up here. They're not super easy to see, but there are definitely some hints of spindle fibers right there. Here's that invisible metaphase plate. The chromosomes are lined up across the width of the cell here, and there's little spider legs coming off of that center line. This cell is in metaphase. And that brings us to anaphase, which is another very distinctive phase when you look underneath the microscope. This is the stage where the action starts. The sister chromatids are literally going to be pulled apart as the spindle fibers start to retract, start to shrink back. Individual chromosomes will be pulled then away from their partner's chromosome, away from their sister, and to opposite poles of the cell. Now your textbook shows it like this. It shows it like these little V's being pulled away from the center. This set will be pulled up to this pole, and this set will be pulled down to this pole. Now, the reason the chromosomes V like this is because the spindle fibers are attached to the kinetochore, the little black dot. And when the spindle fibers start to shorten and retract, they're pulling at that black dot level. So the rest of the chromosome, the ends of the chromosome, literally sort of lag behind and create this V pattern. The tips of the V's on this side are being pulled to this pole, and the tips of the V's on this side are being pulled to this pole. The spindle fibers here are literally shortening. And this is what it looks like under a fluorescent microscope. I just think these images are amazing. You've got green spindle fiber staining in this picture and blue chromosome staining. Over here, green spindle fiber staining and blue chromosome staining. So it's very clear in this picture that we're moving away from the center, which is right here, and we've pulled the pairs of chromosomes apart. This set is going to be pulled in this direction, and this set is going to be pulled in this direction. And this is what it looks like underneath the microscope. This is labeled as early anaphase. We would call this anaphase in our course. You can see that they're no longer lined up along the center of the cell. There's space here. we've got two sets now. We've got one set here and one set here. This set is being pulled towards this end of the cell, and this set is being pulled towards this end of the cell, and that's anaphase. This is an even nicer image. This is a little higher power. We're still looking at a plant cell. You can see the very rigid cell walls here. We've got a centrosome up here somewhere that we can't see. We've got maybe the hint of spindle fibers. We've got a set of chromosomes here. We've got another set here. This set is being pulled to this side. This set is being pulled to this side. And you can really see the V shape in this image, the V shape of these chromosomes. So let's take a look at this slide, which has, again, a section of onion cells, just like you'll see in lab. And this is a nice section because it's got several different mitotic figures in the same view. So if I pointed at this cell nucleus, or this nucleus, or this nucleus, what phase would you say these cells are in? If you guessed interphase, you're right. This dark red structure is a nucleolus, but all of this nice even looking material in the rest of the nucleus tells me this is interphase. This is interphase. There's a nucleolus. This is interphase. There's a nucleolus. How about this cell right here? This cell is in metaphase. The chromosomes are lined up across the center, even though there are some spider legs sticking off of it. This is metaphase. How about this one down here? That one would be called anaphase. We've got one set here, being pulled in opposite directions in the cell. Now, I could probably point to this one or perhaps this one as examples of prophase. I say that because there's a lot of speckling in the nucleus up here and over here, which tells me that the chromosomes are beginning to condense. So I would call this a prophase, and I would call this a prophase. Good. Now the last stage of mitosis is telophase. Think about what you have inside the cell at this point. We've got one set of chromosomes pulled to one end of the cell, one set of chromosomes pulled to the other end of the cell. The next thing that has to happen is we have to recreate nuclear envelopes around those sets of DNA. So the nuclear envelope is going to reform during telophase, and the spindle apparatus, all of the spindle fibers, are going to break down and disappear again. The spindle apparatus is literally going to disintegrate. What this means is that during telophase we have a single cell that has two nuclei in it. Very unusual. And this is the way your textbook depicts it. One cell that now has two fully formed nuclei in it. And the other thing you can see, of course, is that the spindle is starting to disappear. It's starting to break down because it's no longer needed. It's already performed the work it needs to do. And this is what a telophase stage cell would look like under the microscope. Telophase is hard to find under the microscope, and it may be one that we're searching for on Thursday. But I think you can make out that this is one cell. I can copy around the cell wall. I can trace around the cell wall here pretty easily, and I can see two groups of chromosomes in here. Now the reason I wouldn't call this anaphase is because I'm starting to see a very circular shape to these chromosomes. Instead of seeing all those upside down V's, I'm seeing instead what looks like a new nuclear envelope forming. So I would call this a telophase cell. Here's another one. I've got a cell that I can outline here. One cell with two nice rounded brand new nuclei in it. I would call this telophase. Now, because it's a plant cell, I'm not going to see a pinching off of cytokinesis. Instead, the plant cell is going to lay down new wall right here. And you can just start to see that new wall forming if you look very, very closely. This is a cell that's in a very late part of telophase. I know that because look, this is the brand new cell wall that has formed. So a short time ago, this purple line wasn't here, and we had two nuclei inside one cell. The reason I know that these aren't just two cells sitting side by side is because I can still see those nice anaphase chromosomes sitting inside a brand new nuclear membrane. If this were just an ordinary cell in interphase, let's say, then all of this would just look nice and homogeneous. I wouldn't see these very distinct, lined up sort of spider legs inside the nucleus. All right, so let's take another look at the video that we looked at the very beginning of the lecture. Remember, what you have here is nuclear material that has condensed, and we can see individual chromosomes inside the nuclear envelope. I got a nice rounded nuclear envelope around these chromosomes. I've got spindle fibers starting to form out here. I just can't see them. When I set it in motion, you'll start to see things moving and shifting. The nuclear envelope is going to break down for prometaphase, and the chromosomes will be set loose into the cytoplasm, right about there. Then they're going to come and line up in the center for metaphase, and I'll tell you where I would call it metaphase. Maybe right about there. Nice straight line across the middle. Now watch, it'll happen fast. We'll go into anaphase as we split the sisters. We pull one pair this way and one pair this way, anaphase. Then we go into telophase, and we're going to wrap a brand new envelope around each set of chromosomes. The chromosomes will decondense, they'll go back to normal. We'll put a new, brand new cell wall in place, and we'll be finished. This is a short video that I found on the internet that I thought was great for this material. This must have been someone's project in biology class. This is a human dramatization of mitosis in a swimming pool. So, take a look. What we have is a cell. We have chromosomes inside the cell, and what we're going to have happen now is they're going to line up in the center, and they're going to attach to the pool noodles, which are our spindle fibers, and then our two spindle apparatus people are going to pull the sister chromosomes apart. In just a second you'll see that happen. Anaphase. Here comes our brand new cell wall jumping in the pool, and now we have one nucleus here, one nucleus here, and a brand new cell wall. I thought that was pretty cute. Okay, so that's the end of the material for mitosis. Remember, when we look under the microscope on Thursday, the vast majority of cells that we see are going to be in interphase. Each cell is going through the cell cycle at its own pace. In other words, they're not all synchronized. They're not all going through the cell cycle at exactly the same stage. So, just by the law of averages, because interphase contains G1, S, and G2, most of the cells will be somewhere in there. And our job will be to hunt out the cells that are in mitosis, or M phase, and to see what kind of mitotic figures we can identify. can we find prophase, can we find metaphase, anaphase, and telophase. That will do it for today's material, and we'll see you in lab on Thursday.